Hello, my name is Dr. Jeremy Petrovich, Developmental Editor for Current Protocols at Wiley. It is my pleasure to introduce Dr. Julie Burrell, a member of the research faculty of the Peters Laboratory located at the Vaccine Discovery Division of the La Jolla Institute for Immunology. Dr. Burrell received her PhD in immunology in 2015 from the University of Queensland's Brisbane, Australia. Following the completion of her graduate studies, she accepted a postdoctoral fellowship under the mentorship of Dr. Bjorn Peters at LJI in 2016 and has been recently promoted to the level of instructor research faculty in early 2020. Dr. Burrell's postdoctoral studies have been primarily focused on systems immunology approaches by combining multidimensional data sets such as flow cytometric data and transcriptomics to understand inter-individual immune variability and to find new correlates of protection to infection and vaccination. Dr. Burrell has more than 10 years of continuous training as a human immunologist with extensive experience in flow cytometry, including multi-parameter flow cytometry, cell sorting, and imaging flow cytometry. Her contributions to the field of flow cytometry include the development of universal frameworks to standardize flow cytometric experiments across research centers, and more recently the discovery of a novel immune cell-cell doublet population that can be detected by flow cytometry in human blood and is more prominent in individuals with active disease, indicating clinical relevance. So with that, I will turn this webinar over to Dr. Burrell to present a talk titled ImageStream and the Challenge of Distinguishing Cell-Cell Complexes from Singlet Cells in Flow Cytometry. Thank you.
Thanks, Jeremy, for the very kind introduction, and thanks everyone who has joined this webinar. It's my pleasure to be here today and telling you about cell-cell complexes and how actually we can find them, deal with them, especially using the Amnis ImageStream technology. So I will start by a bit of a history of how did we came across this cell-cell doublet population. So our lab is really interested in doing some cell subset transcriptomics in disease. And in particular, we are a lot interested in T-cell subsets. So in this experiment, we started off from frozen PBMC from either TB uninfected or latent tuberculosis infected individuals. And so we sorted memory CD4 T cells as shown in the gating strategy here. We gated on lymphocytes, excluded doublets as usual, then got the live CD3, CD4 positive T cells, and then detected the memory based on CD45RA and CCR7 expression. And we then sequenced, RNA sequenced this population, and we found a very nice gene signature that can discriminate between latent tuberculosis and our control. But then when we started looking a little bit further into the variability within the data set, we found that looking at the 1,000 most variable genes, as you can see in this heat map here, a set of 22 genes that were really highly co-expressed with each other, which are highlighted in this red box here. So the variation was great amongst individuals, but within the same individuals, all these genes would be highly co-expressed. And this doesn't seem to be related to the state of latent tuberculosis, as you can see that both control and latent tuberculosis individuals can have or not these genes. But what was really more striking is that when we looked at the nature of these genes, we found that they were mostly monocyte genes, and in particular we had the lineage marker for classical monocyte CD14. And so none of these genes actually would be expected to be expressed in T-cell subset, while here we had sorted memory CD4 T cells. So we decided to look back into our FCS file from the sorts. And so we looked into, after gating on CD4 memory T cells exactly the same way as was doing during the sorting, can we actually find some CD14 positive events? And indeed, we found that in gated CD4 memory T cells, there was a small population of CD14 positive events. And then if you see on the graph on the right, the frequency of these CD14 positive events within sorted CD4 memory correlates perfectly with these 22 variable genes, which were monocyte genes that we observed, which means that this CD14 population is likely responsible for this monocyte gene expression that we detected. So we decided to gate back from the CD14 positive cells and see within our overall gating strategy, could we actually flag these cells as being different from any other memory CD4 T cells. But as you can see here in the plots in the bottom in red, all the CD14 positive memory CD4 T cells are actually looking exactly similar as other memory CD4 T cells. They have the same expression of CD4, of CD3. They are not staining more positive for the live dead stain. They fall as well into the singlet gate and in the lymphocyte gate as well. So we decided to take a step further back and actually look within the overall cells, this time not looking only at CD4 memory T cells, but the overall population by getting out all the doublets or aggregate, taking the live cells. Can we actually see a population that double express CD3, which is a T cell marker, and CD14, which is a monocyte lineage marker? And indeed, we found in every sample that we analyzed this double positive population that could also be subcategorized into two populations. So we have in blue here the CD3 positive with a CD14 high expression, and then in red a CD3 positive T cell population that also have a mid expression of CD14. And if we backgate on forward and side scatter for these two cell population, we can see that the CD3 positive CD14 high in blue is actually mostly associated with a higher forward and side scatter, which are usually typically where monocyte lies. And then the CD3 positive CD14 mid population in red would be mostly where the lymphocytes are, and that's exactly the cell population that was sorted along with our other memory CD4 T cells and responsible for that monocyte gene expression that we observed. So this is where we decided to turn into the ImageStream technology to actually have a look on what we were gating on. And so we used the Amnis ImageStream and the same gating strategy that we applied to identify the CD3 positive, CD14 positive cells using the BD-ARIA cell sorter. And so I'll come back later in the presentation on the exact gating strategy that we used, as you will see that it's actually critical to get the proper interpretation of what you're seeing in non-imaging flow cytometry. But so here, the beauty of ImageStream is then once that you gate your population, you can actually visually export gallery of events and see what you're actually gating on. And so when we looked at what we had in our CD3 single positive or CD14 single positive, as expected, we had single cell that would either express CD14 for the monocyte here in red, or in the T cell population, we had singlet cells that would express CD3 uniformly on the cell surface. And then to our surprise, when we looked at CD3 positive, CD14 high population, instead of finding one single cell that would double express CD3 and CD14, instead we found a doublet. So a T cell and a monocyte in a very strong interaction with each other. In actually such a strong interaction that they were not flagged as a doublet using the typical strategy in non-imaging flow cytometry using the A versus H or H versus W from the forward and the side scatter. When we looked at what was the CD3 positive, CD14 mid population, we found that it was mostly singlet T cells with either a monocyte that would have a dimmer expression of CD14, so maybe a non-classical monocyte. And those that were not doublet but singlet only often had a sort of fragment of CD14 here in red along on their membrane. And so when we looked a bit closer at this cell fragment, you can see them on the left by ImageStream flow cytometry. It really looks like pieces of monocyte that are still attached to the membrane of the T cell. And by confocal microscopy, we also confirm that this size is actually way bigger to be any like exosome or microvesicle, but in what seems to be really a cell fragment. So here we think that potentially they were also T-cell monocyte doublets, so complexes together. But here during the acquisition, actually, the interaction was torn apart, the monocyte lysed, and some actually still was retained on the membrane of the T cells. So this is a sort of hallmark of a previous interaction. So this was all done with imaging flow cytometry. So then we wanted to confirm that what we saw here was actually matching with what we were having in the BD ARIA cell sorter. So we directly sorted CD3 positive, CD14 positive cells along with single monocyte or T cell in a slide, and then ran into the microscope to actually look at what we had. So the cells are, of course, not in a very good shape, as they are just sorted directly into a glass slide. But you can see here this crystal form, which are actually the sorting buffer. And then we could find in each of the single cell monocyte that was sorted, we could always find within the crystal one reddish looking cells that would be the monocyte. It has a little bit of green as its autofluorescence for that fluorochrome that was used for CD3 as well. And then for T cells, we always could find one very bright green event, which was that single T cells. But then for the CD3 positive, CD14 positive cells, we could always find actually two cells. So what the cell sorter was actually considering as being only one cell was actually containing two cells, one T cell and one monocyte. So this is not the first time that cell-cell doublets are being reported to be present in human samples and in flow cytometry, but usually they are known to be or believed to be the results of technical artifact. And this would be discarded in the data analysis or even not acquired at all, as usually we use our gating strategy to exclude them. So in order to rule out that actually these were results from technical artifact, we compared, also all of our previous work was done on frozen PBMC. And so one could argue that actually the cryopreservation would create artificially these cell-cell doublets. But so we compared side by side from the same sample, either fresh or frozen PBMC, the frequency of these T-cell monocyte complexes, and we found a very nice correlation, ruling out that the cryopreservation could be responsible for their formation. And then more strikingly, we also looked directly from whole blood, just by depleting magnetically red blood cells, and we found again a very nice correlation with the frequency of T-cell monocyte complexes present in the fresh PBMC sample. So you can appreciate here that there is actually about a twofold difference, where in the fresh PBMC you have about twice more T-cell monocyte complexes than in the whole blood, which seems to show that actually the phycal preparation might create some, but that you have already a significant fraction of T-cell monocyte complexes present in the whole blood at the time of blood draw, which are this likely to be present in vivo in the individual at the time of the blood draw. We also looked into how stable over time these T-cell monocyte complexes are. So we took we selected 5 healthy individuals, and we collected PBMC 1 week apart, and then looked between the 2 visits in frozen or in fresh PBMC how many T-cell monocyte complexes we can find, and we found a very strikingly stable frequency. So all of this to us rule out that those could be responsible for technical artifacts during ex vivo sample manipulation, but that actually these T-cell monocyte complexes are present in vivo in the individuals. We were there interested to see whether we can disrupt their interaction. And so we did different chemical or physical treatment. And so we found that adding more EDTA didn't change the frequency of T-cell monocyte complexes. If we start going into a bit more harsh reagents, such as using a red blood cell lysis reagent, we started losing some of them. But that's really by going more through a physical manipulation of the sample, such as pipetting very vigorously up and down for about 30 seconds, where we start actually breaking them down And then the sonication was the most efficient, a mild sonication, of course, making sure that most of the cells were still alive. So then we were looking at what potentially could be the mechanism of formation and how are they actually attached to each other. And so one of the very well characterized immune interaction between T cell and monocyte is actually the formation of immune synapses, as monocyte would serve as an antigen presenting cells. And so usually upon activation and contact, you have a lot of restructuration of adhesion molecule. On the T cell side, usually you have LFA1, and on the monocyte side, ICAM1, that would all polarize at the point of contact and form this sort of ring-like structure that can be seen by microscopy. And so we isolated some of the T cell monocyte complexes and then looked for this adhesion molecule. And as you can appreciate here in the example shown, you can see a very nice polarization and sort of ring structure of both LFA1 and ICAM1 in the doublet that we isolated. And so we found this polarization in about 30% of the doublets that we've analyzed. It's going, again, matching the results that we found in the whole blood versus fecal, where we see that the fecal would actually create some, and so we think that the one present directly in the whole blood are probably likely the one that will have this polarization and really be a hallmark of this in vivo interaction. So then we were interested to see whether every type of T cell or not can be found in a complex with the monocyte. So when we looked at CD4 versus CD8, we found that indeed each different T cell subset can be found in a complex with the monocyte. And we also found that the naive versus central memory or effector phenotype, every of these 4 different subsets could be found as well in a complex with the monocyte. And so then the key question that we wanted to address was, as actually the system is getting perturbed, so for example, someone is getting infected, then are there an increased frequency of these T cell monocyte complexes, as if they are actually indeed a hallmark of immune cell interaction? One would expect that when the system gets perturbed, then you would have an increase of immune cell interaction, and so maybe an increase of T cell monocyte complexes formation. And so one thing that we found by analyzing a lot of our samples is that actually, not very surprisingly, the frequency of T cell monocyte complexes was directly correlated with the frequency of the individual component of the complex. So if an individual has more T cell and monocyte circulating, then the likelihood that they will be in random association increase. And so to correct for that, we decided to use a metric called that we named the Ka, so for constant of association, and a little bit like in chemistry, where we divide the frequency of T cell monocyte complexes by the frequency of the individual component, therefore correcting for this. And so then we looked at the T cell monocyte complex constant of association across different immune perturbation models. So we first looked into active tuberculosis, and so we had longitudinal samples of individuals at diagnosis and then 2 months post-treatment. And we found that there was an increased frequency of T cell monocyte complexes at diagnosis compared to 2 months post-treatment. In the case of a viral infection, such as dengue, we found that individuals with a more severe form of dengue fever, the dengue hemorrhagic fever, had a higher frequency of these T cell monocyte complexes compared to previously infected individuals. And then finally, in a vaccine model, using individuals that receive the tetanus, diphtheria, and acellular pertussis boost, we found that 3 days post-vaccination, you have an increase in the T cell monocyte complexes constant of association compared to before immunization. So it seems that indeed, as the system is getting perturbed, potentially you have more immune cell interaction, and so you find more of these T cell monocyte complexes in the blood of the subjects. And so then more strikingly, we also looked at what type of T cell is actually present in association, as if it's indeed reflecting in vivo immune cell interaction, then it's expected that for different pathogens, you would actually elicit different qualitative immune responses. And so we found that in the case of active tuberculosis, which is a bacteria, is expected to be both intracellular and extracellular, but majorly it's known that CD4 T cells are quite a strong component of the immune response. And we found indeed that the constant of association with the monocyte for CD4 was higher than for CD8. For dengue fever, on the other hand, it's a virus. So here it's expected more to have strong CD8 T cell response. And we found that the ratio of CD8 over CD4 was more biased this time towards CD8. You had more CD8 T cells in a complex with monocyte. And then finally for pertussis, which is an acellular vaccine, here we expect very strong CD4 T cell responses. And we find indeed a much higher constant of association of monocyte for CD4 compared to CD8. So here we are showing that on top of being a differential quantity or frequency in the blood, also the type of T cell that is in preferential association with the monocyte will vary according to the immune perturbation and reflect actually the expected polarization of immune responses. And then the other observation from this data that we can make is that the constant of association for double negative T cells actually was the highest in all three models. And so maybe the double negative T cells, which are probably predominantly gamma delta T cells, have a higher affinity for monocyte or a more likelihood to be present in a complex, maybe through a new mechanism of adhesion that haven't been described yet. Next, we wanted to see whether we could generalize all of these findings to any type of T-cell APC, as indeed T cells are able to form this immune synapse and complex structure with any antigen-presenting cells. And so we took advantage of our Tdap boost vaccination longitudinal model, where we had 10 individuals that were given the vaccine, and then we followed over the first 14 days the frequency of CD3 positive, CD14 positive event, which are T cell monocyte complexes. And so as I've shown already before, you have an increase at day 3. But then, interestingly, we also find some CD3 positive, CD19 positive event, which we think might be T cell, B cell complexes, and then CD3 positive PDC or CDC events, and all of them picked at about day 3 post-immunization before going back to baseline. So we think that all of what we've just described could also be actually applied to any T-cell APC doublet. So now that's it for the introduction of what are cell-cell complexes, why are they present, and what do they represent actually, probably through in vivo interaction. But now I'd like to take the remaining of the webinar to actually discuss what are the implications of this doublet hiding in the singlet gate for single-cell technology. So if you don't know that you're actually facing a case of doublet, if you just look, do a flow cytometry analysis of the CD3 positive, CD14 positive cells, well, then they will look, for example, if you look at monocyte canonical marker, because they actually contain a monocyte, an entire monocyte, they look exactly similar to a monocyte. And then the same if you look for a T-cell canonical markers, they will look exactly like a T-cell. So then if you start integrating all of that together and do more high-dimensional analysis, here, for example, it's a cytoph analysis of CD3 positive, CD14 positive cells, then they will look exactly like a hybrid mixed population between T-cell and monocyte. So then, if you decide, okay, maybe then here I've discovered a new cell type that is a mix of T-cell and monocytes, that's at the protein level, so then you will want to go and say, well, let's just single-cell sort this and analyze it at the RNA level. But then again, because in the single-cell sort, it will contain 2 cells, then you will have sequencing profile that will look really similar to a T cell and to a monocyte. And so by integrating all of that together, it will look like a separate mixed population in between T cell and monocyte. So here we can understand that it's very important to be able to flag that there are potential cell-cell complexes in your single-cell data. Otherwise, you might have complete new interpretation of saying that you've discovered a new hybrid cell type, while actually you've just discovered a complex pairing one T cell and one monocyte, but it's not a new immune cell type per se. So we've been therefore interested to see how can we better discriminate cell-cell complexes from singlet, as it seems that the non-imaging flow cytometry technology actually failed to flag these as being doublets or aggregates. So of course, if we looked at imaging flow cytometry, it's a very powerful tool to actually discriminate between singlets and doublets, because it has actually a camera that can take a picture of your cell, and so you get this key bright field parameter that is lacking in non-imaging flow cytometry. And so by deriving the area and the aspect ratio from this bright field parameter, you can very thoroughly discriminate between singlets and doublets. So you can see here that the monocyte population and T cells are mostly just entirely singlets, where the CD3 positive, CD14 positive population really fall into this doublet gate in red. And so you can derive actually and calculate metrics for that by doing a ratio of the area versus the aspect ratio. And so you can see that it's very high for the CD3 positive, CD14 positive population, flagging it very nicely as a doublet. And then if you want to see whether actually is it a doublet of two cells that would co-express potentially CD3 and CD14, you can also use the area versus aspect ratio, not only for the bright field, but also for the each of the fluorescent parameters. So here CD3 and CD14. And you can see here that the area versus aspect ratio go down to being the level of a singlet, showing that here you have a doublet, but that contains one CD3 and one full CD14, and not two double expressing cells. So that's another sanity check metric to look in your imaging flow cytometry data. Another very helpful marker to discriminate between singlet and doublet is just ERGT, that will actually look for the nuclei of cells. And so here you can really nicely see this double population for CD3, CD14 positive cells. And if you sum up the MFI obtained from ERGT, you actually have a perfect, your T cell monocyte complexes will be the perfect sum of T cell and monocyte. So that was all for imaging flow cytometry. But what if you don't have access to it? Or what if you actually want to sort your cell population? We have yet to have an imaging flow cytometry that can sort cells. So at some point, you will always have to translate this finding back to the non-imaging flow cytometry. And so we were interested to see if in non-imaging flow cytometry, can we have parameters that can discriminate between these complexes from singlet. So if you look here at the forward and side scatter, you have your T cells that are lymphocytes, so usually smaller forward and side scatter, and then the lymphocytes, which are associated with a bigger monocyte forward side scatter in red. And then we have the T cell monocyte complexes in blue, and you can see a very strong overlap with the monocyte population. So unfortunately, the non-imaging flow cytometry is not enough. We cannot actually draw a gate to completely encompasses the complexes without taking some of the monocyte or even some of the lymphocyte. But if you derive the mean fluorescence intensity for forward or side scatter, you can clearly see that it's higher for the T cell monocyte complexes population. So that's just a very simple sanity check to do whenever you have a cell population that is dual expressing for two lineage markers that you would not necessarily expect. You can just gate back and have a look that this cell population have just a higher forward and side scatter than my other population. And then another very useful marker to us was CD45, as it's expressed by every leukocyte, so both monocytes and lymphocytes. And so when we looked at the mean fluorescence intensity for CD45, we found that it's increased for the T cell monocyte complexes because they actually contained two cells. So those were really useful to us in non-imaging flow cytometry, a very simple sanity check to do in any suspicious dual expressing cells that you might encounter. And so then if you as well undergo single-cell RNA sequencing, you can also go back to your single-cell RNA-seq data and see some hallmark of cell-cell complexes. So what is very interesting with RNA sequencing is that at the difference of flow cytometry, that is an absolute quantification, here you have more a relative quantity of mRNA. And so each gene that is actually expected to be only in one cell type but not the other, in the T-cell monocyte complexes, because you have the double amount of mRNA, actually the level of this gene would be about halfway than in the single population. So here, if you look at monocyte genes, in T-cell monocyte complexes, all these genes are about half the level that would be in single monocytes. And so that's because of this normalization. And again, the same can be seen for T-cell genes that are about halfway. So whereas in the heat map, it's not necessarily visually, you can't really see it. You can see here a little bit that the yellow, which is so the high expression, is a little bit dimmer for the T-cell monocyte complexes. Just by summing all these TPM together, it appears way more significant. And so that's a very, as well, simple sanity check to do on data that you've already generated to be sure that you might not have, you have or not a case of cell-cell complexes. So then if you suspect that you might have a cell-cell complexes, then usually you will want to then image. And so that's where imaging flow cytometry can be very useful. And so here I'm going to talk a little bit about the importance actually of checking for the right gating strategy using this non-imaging and imaging flow cytometry platform. So here I'm representing the gating strategy for T-cell monocyte complexes, so the CD3, CD14 positive population. So as I mentioned before, we just gate on cells. We exclude doublets or aggregates, but actually now we know that it doesn't remove entirely the doublets, and some will still be retained into these gates. We take the live cells, and then we get our CD3, CD14 double positive population. So then what do we do if we are in imaging flow cytometry? We don't have access to forward scatter, but there you have access to the bright field. And so usually we would use the bright field area, the side scatter, and then you could very nicely identify your cells by removing the debris and the aggregates. And then, as I've mentioned before, you can use this bright field area and aspect ratio to really nicely identify your singlet cells. Then you take the live cells, and you get the double positive CD3, CD14 cells. But as you can see here, it looks like we have a way lower frequency of events compared to the non-imaging flow cytometry. And so that was seen for all of the samples that we tested. We always obtained a lower frequency. So it means that potentially this gating strategy actually is not matching really what we are seeing in non-imaging flow cytometry. And so we hypothesize that this is because the bright field actually parameters is a way more powerful way of identifying, more accurate to discriminate doublets from singlets. And so we were more interested to play a bit around with different gating strategy and found that actually by dropping this bright field exclusion of doublets, we actually could reproduce better what we're seeing in non-imaging flow cytometry. So in the end, we came up with a new gating strategy that we called a cell sorting matching gating strategy, where this first step of just taking cells and no aggregates actually was sufficient to reproduce the first three steps in non-imaging flow cytometry of getting cells and then removing the big aggregates and doublets. So then by taking live and CD3, CD14 positive, we could again find the same matching frequency. So we think that this cell sorting matching gating strategy is a way more accurate way to really see what you were seeing in the cell sorter and so have a better idea of what you were actually sorting. And so if we look at these two different gating strategies, actually they will give you very different results on interpretation on what is your double positive cell population. So here, if you use the cell sorting matching one, the one that gives you the same frequency of events across the two platforms, you find that indeed CD3 positive, CD14 positive cells are not singlet, but they are a complex, a doublet with a T cell and a monocyte. However, if you use the expected or optimal imaging gating, then this time you have a way fewer events, and those events are actually singlets that do co-express CD3 and CD14. So here it's showing how important it is to match the gating strategy, because if you're not careful enough, then you will do imaging flow cytometry, do this optimal imaging, and then have the interpretation that all of your CD3, CD14 positive cells are true singlets that co-express. Well, actually here we are showing that yes, the single dual expressor cell exists, but they do only represent a very small fraction of your overall CD3 positive, CD14 positive events, and the majority are actually doublets pairing a T cell and a monocyte. So that brings me here to our recommendation. If you encounter case of suspicious dual expressing cell population in flow cytometry, first, if you have data already generated, just quickly check for signatures in your flow cytometry data. Does it have a higher forward and side scatter? If you have CD45, just look at the MFI of CD45. In the single-cell RNAseq data, you can look for genes that are expected to be only in one cell type and not the other. Are they at the same level or halfway? If they are halfway, that's usually a sign that you might have a doublet. And then, if you think that you potentially have doublets, you can start performing imaging flow cytometry. And here, this is key to actually use an appropriate gating strategy to get the same matching frequency of events that you would see in the non-imaging flow cytometry. And so then another experiment that to us actually was really helpful was to just perform a direct microscopy analysis of sorted cells. So there you don't have the problem of actually translating correct gating strategy between different technology. You just directly visualize what the machine was sorting. And so here this is where we found that when we sorted the CD3 positive, CD14 positive population in microscopy, we had then a perfect stoichiometric mix of CD14 positive cells and CD3 positive cells, but very, very few dual expressors. So this was our first piece of data that started convincing us that, oh, maybe we have a case of complexes, and that's what led us to look at different gating strategy in imaging flow cytometry. Otherwise, we might have completely missed as well this interpretation. And so finally, for the remainder of the talk, I just would like to highlight the potential application of this recommendation and to other type of Tcell APC complexes. So at the time that we published the first paper on Tcell monocyte complexes, came another very interesting paper in Cell that was describing in type 1 diabetes patient a novel cell population with both T-cell and B-cell lineage specificities. And so the author renamed these cells DE cells for dual expressor cells. And very importantly, these cells encoded for a very potent T-cell autoantigen that could be involved in diabetes pathogenesis. And so at the time that the paper came out, a lot of our collaborators, actually, which were aware of our cell-cell complex work, were also wondering if this work could be impacted or not by maybe T-cell, B-cell complexes. So this is here how they gated for DE cells. So they looked for CD5 and CD19 double expressing cells, and then they looked for TCR alpha beta expression and IgD or not. And so then they found that in type 1 diabetes patient, you have these CD5, CD19 TCR alpha beta positive cells at much higher frequency compared to healthy individuals. So they then performed single-cell RNA sequencing of these cells and found that they did express both B cell and T cell markers, as you can see here in these heat maps. And they also had their own gene expression signature on the top panel on the heat map. And then they performed as well, of course, imaging flow cytometry and shown very convincing dual expressing cells that had both TCR and immunoglobulin receptor on the surface. So the flow cytometry data was not available, so we decided to just try within our healthy PBMC sample, can we identify this cell population? So we used the same gating strategy as they did by taking cells, excluding through A versus W the singlets. By tipping just the singlets from the forward and the side scatter. Then gating on live cells, and then CD5, CD19 positive, and then found this TCR alpha beta positive cells. And so when we looked at our just quick flow cytometry check, we found that these TCR alpha beta positive, CD5, CD19 positive in blue actually associated with a higher forward and side scatter, as well as also higher CD45 expression. So they seem to potentially contain cell-cell complexes. We then looked at their single-cell RNAseq data, which was nicely publicly available. And so when we looked at B-cell genes or T-cell genes, we found that in DE cells, all these genes were about halfway the level as in single B cells or single T cells. So in our hands, this was again showing that maybe this is here a signature of T-cell, B-cell complexes that were sorted. And most strikingly, when we looked at the specific signature of DE cells, we actually found a high overlap with our 22 variable genes that we found initially in memory T cells that were monocyte genes. And in particular, there is the lineage marker CD14. So we think here that maybe on top of containing T-cell, B-cell complexes, there might be some monocytes as well present in this cell population. So why is the case of D cell might not have been seen by this study of Ahmed and al. For D cells? So they've done really proper analysis and good imaging flow cytometry. And so when we tried to look at the gating strategy that was used, the information that we could find from the methods was that they used an aspect ratio that was consistent with single events and devoid of debris or multicellular events such as doublets. So to us, it seems that the strict, optimal gating strategy was used here, where you actually use the bright field parameter, but we've shown that actually this is not really matching what you are actually sorting. And so when we apply this to our samples, we try to do to identify D cells with the cell sorter, and then with the imaging flow cytometry using different gating strategy, we found that when we use this optimal imaging, you find a much lower frequency of events. And then if you look at the gallery with this optimal imaging, yes, indeed, you find singlet cells that co-express TCR alpha beta and CD19. However, when you use then the cell sorting matching one, so the one that gives you the same frequency, so more likely what you're actually detecting in your sorter, you find that you have mostly T-cell, B-cell complexes. And so here we think that the dual expressing cells indeed exist, and they might actually be really more prevalent in their samples as well, because here we are looking at our own healthy samples. But they might potentially represent only a very small proportion of what you actually sort. And so then the whole interpretation that you have a new immune cell type at both protein and RNA level might not hold true. And maybe they are only dual expressor at the protein level and have acquired this marker by trogocytosis, for example. So that brings me here to the conclusions of this talk today, where I hope that I've convinced you that cell-cell complexes actually are present in the single gate of human PBMC. And so in particular, we looked at the one pairing a T cell and a monocyte, and that they are importantly not technical artifacts as usually believed in the field of flow cytometry, but they do reflect Tcell APC in vivo interaction. So actually by looking at them, you can get key information on the immune cell types that were currently interacting and communicating with each other at the time of blood draw. And so these cell-cell complexes, because they are not flagged as being a doublet by non-imaging flow cytometry, they will appear at first glance in your data analysis to be a halfway cell population with mixed lineage features at both the protein and the mRNA level. And so we recommend to look for in-flow cytometry and single-cell RNAseq data just for these simple molecular signatures that we highlighted, just simple parameters to have a look at. And then we highlighted the importance of electing the appropriate gating strategy when using ImageStream to actually accurately reflect what is the dual expressing cell population that you detected and sorted in non-imaging flow cytometry. And then by applying this knowledge to other type of Tcell APC complexes, we found that a recently described novel cell population with both Tcell B-cell lineage property might actually be affected by the presence of Tcell B-cell complexes. And this doesn't change at all the whole conclusion of the paper that they found a potent autoantigen. It's just that maybe it's not a new cell type, but it's just a complex of a T-cell and a B-cell. And so until high throughput imaging cell sorter is actually being developed, we need to systematically check and report in single-cell immune profiling study the presence of cell-cell complexes, especially for those that report on the discovery of novel immune cell population. And so most of this work has been published. So in eLife is our first description of T-cell monocyte complexes, and then more the technical challenge of distinguishing cell-cell complexes from singlet is in bioRxiv and should be out really soon in cytometry part A. So with that, I'd like to thank everybody that has been involved with this work. It was a very fun, and it's still a very fun collaborative project that involved a lot of different core and technology, going from clinical core sequencing, bioinformatic, flow cytometry, and microscopy. And all the imaging flow cytometry work was done at Stanford Burnham with the help of Yoav Altman. And I'd like to thank Luminex for giving me this opportunity to give this webinar today. And thanks, and I'll take any questions.
Thank you, Dr. Burrell, for that presentation. For questions from the audience, one of the questions is, are Tcell APC complexes enriched for antigen-specific T cells?
Yes, so that's a good question. I haven't presented that today as a matter of time, but we started looking actually by TCR sequencing because we found an enrichment for immune synapses. We were thinking that maybe we would find enrichment for antigen-specific TCR. And this was only a very preliminary experiment, but it didn't really find any enrichment so far. So that's something that we are pursuing. So since we know that maybe not all of them are in current immune synapses and in fully mature immune synapse, it might be actually another type of cell-cell interaction, which might not be necessarily antigen specific.
Have you been in contact with the authors of the Cell paper regarding your divergent interpretation of the nature of DE cells?
Yes, we have. And so we've been communicating quite a bit back and forth by email when we gave them our interpretation. And they acknowledged our vision, but they still think that they have a case of true dual expressors. And the reason might be that what they've used in their paper is the use of IgD. And in the end, for our gating strategy, we only looked at CD5 and CD19 and TCR alpha beta, but we never looked at IgD. So they think that's why maybe we differ, and for our case, that was only T-cell, B-cell complexes. However, to me, that still doesn't explain why there is single-cell RNAseq data which is derived from their sample, to me also have a signature of complexes. But I hope that we can correspond after our paper is being published, that we can just exactly openly correspond about different interpretation. I think this is how we can advance science.
I definitely agree. Any clues on mechanisms for the formation or adhesion of cell-cell complexes, and have you looked into inducing their formation in vitro?
Yeah, so that's a question we have quite often. So we tried. Unfortunately, we haven't been successful so far. And it's probably due that monocytes are very sticky and adherent, so it's kind of hard once you put them in culture to get them back. We might have more luck with T-cell, B-cell, but so far the mechanism of formation are quite still unknown, as well as the adhesion. As I mentioned, it's not a fully mature immune synapse. We have a lot of double negative T-cells. So we are currently doing some quite deep RNA sequencing on T-cell and monocyte in a singlet versus doublet to actually see whether we could see a whole bunch of adhesion genes that would be upregulated in cells in doublet versus not, that could give us clue. So we hope we will have maybe some answers in the next few months.
A follow-up to that is, have you looked for cell-cell complexes in animal models, and have you actually found any yet?
So we had one collaborator that did try in the mouse model, and so they could identify some. They looked specifically, they were really trying to match what we were doing, because here all this work is done on human blood cells, and so they could find them in the mouse. However, when the system was perturbed, it was not clear whether they were increased or not. And I think here it was more like technical issues of dealing with very small amount of blood in the mouse model. But they've also been reported a lot in tissues, so in animal tissues as well as human tissues. So since our first publication on cell-cell complexes, we've been approached a lot by collaborators or even other people that would tell us, oh, we found this myeloid signature into our sorted T cells, or vice versa. And so then often when we dig in, we actually discover that there is a cell-cell complex. So I definitely think that they are present in animal models and in tissues as well, not only in the blood.
Thank you. Do you think it is more difficult to exclude doublets using classical gating strategy by FACS when the two cell types have different sizes, like monocytes and T cells?
Actually, I think maybe it might be easier, because the bigger the cells are, then the bigger the forward side scatter might be. So in the end, T cell, B cell complexes, because there are two small cells as well, will show then like just a slightly bigger forward and side scatter. So I think it depends. Overall, it's just quite challenging, and I'm not saying that the conventional flow cytometry is not working well. It's more that we still should use forward and side scatter and H versus W, A versus H, all of those. But just keep in mind that you will remove most of the aggregates and your doublets, most of them, but some of them will be retained, and those that are retained might actually be the one with a high biological significance, as enriched for what was interacting in vivo. So I think we don't have enough, like, to answer that question, we don't have probably enough data yet of different type of Tcell APC to see whether it's more difficult or not when it's a monocyte or a B cell. It seems to me to be the same so far.
Thank you. When negatively sorting out CD14 positive cells and then repeating RNA-seq analysis, are the monocyte gene signatures eliminated?
That's a good question. So in theory, so we haven't actually never tried that. And actually that's how we discovered them for the first time, because we forgot to exclude CD14 positive events in our gating strategy, thinking that by just taking the small forward and side scatter, we would get rid of all the monocytes. So I think in theory, if you remove CD14, positive events from your gating strategy, then you should remove that monocyte signature. And actually, that could be a very neat validation experiment to show that your myeloid or your weird gene signature in your sorted cell subset is due to a specific lineage marker. So I would say in theory, yes, it should disappear.
A follow-up to that was, is this doublet population also seen in spleen or in tumors that you've seen?
Yes, so as I mentioned, we do find them in tissues and in some tumor models as well. There's a lot of different inflammation models. So I think any immune perturbation model, when the immune system just has to turn on cell-cell communication a bit higher, then potentially we find them.
So given that this is working with blood samples, can T-cell monocyte complexes be seen on right-stained peripheral blood smears?
So that would be, I'm not sure I'm so familiar with that. I think it's like a smear blood stain. We haven't looked. That would be very interesting. So that would be really straightforwardly take blood and visualize them. I would say in theory we should be able to see them. Probably the challenge is how low frequency they are compared to how many red blood cells do you have. And that's why in flow cytometry we had to magnetically deplete red blood cells before. You need to enrich your white blood cells. So I would say in theory, yes, but I don't know if practically we have the techniques to do that yet.
So to quickly actually circle back on something, for those who don't do a lot of flow imaging in their labs, can you please describe what aspect ratio is?
Oh, yeah, sure. So the aspect ratio is actually on top. So usually we get the area, which is like the entire sort of number of positive pixels to in what the camera snapshot would be, and the aspect ratio is just the height and the width of the signal. So it's to see whether you have this sort of continuum intensity, or if it's like overall cells that express it overall, the marker, or if it's at very focal points. And that would also reflect the size of an event, whether it's a bigger or smaller cell. So it's just another kind of metric to see the shape of your cell and how the fluorescence is being across that surface. I don't know if that was really clear. That's probably information that would be available online if you look for aspect ratio fluorescence parameters.
All right, great, thank you. Our last question is, have you used standard or any specific mask and features to analyze T-cell monocyte complexes in ImageStream?
So any standardization? That was the question.
Yes. Yes. Have you used any standard or specific masks or features?
So we haven't. So the each of the machine, I guess, have been internally standardized. So the ImageStream was run at Stanford Burnham, while we had other flow cytometry like Nonimaging done at La Jolla Institute. So they each have their own, like they are run by specific cores, which have standardized the machine over and over. And then I think here we are just highlighting how we are maybe lacking some standardization protocol actually across different platforms. So I'm not really worried about within a platform that we can repeatedly see the same thing. It's more how do we quickly translate from a machine to another without actually having proper standards of control. So I think this is maybe missing so far.
Actually, we do have time for one more question. Is how strong are the binding affinities of the T-cell monocyte complexes, and is it challenging to image the complexes without breaking them?
That's a very good question. So we found some actually that they were more resilient than we thought, because actually you had to go to like quite physical disruption to disrupt them. But the cell sorting is actually something very stressful for the cell. So once you pass them through the sorter, you will actually disrupt their interaction. So to do all these confocal microscopy and the imaging of the immune synapse, we had to fix the cells, and with quite harsh fixation in order to actually keep them together. And so, of course, there could be the critique that we are actually changing potentially how they were interacting in vivo. So we think it's in between the very strong, it's strong enough that you keep repeatedly seeing them even after you pipette up, down, you centrifuge, you're trying to break down, but it's not strong enough that, not probably covalent, that then if you put them to a thread sorter, a difference of pressure, then you break it down.
All right. Well, we've unfortunately reached the end of our time today for questions. I'd like to thank Dr. Julie Burrell of the Vaccine Discovery Division at the La Jolla Institute for Immunology for taking the time to present to us her talk on the use of the MNIS ImageStream system for distinguishing cell-cell complexes from single cells using flow cytometry and single-cell sorting. Any questions that were not addressed will be answered by email if able. This presentation was sponsored by Luminex in partnership with Wiley's Current Protocols and will be available for on-demand viewing on the Current Protocols website at www.currentprotocols.com in the webinar section under the Resources tab. Thank you for attending.